Now I will show you some important immunosuppressive drugs that are used by the doctors to prevent rejections. The first example is azathioprine. This medical drug is actually derived from, this is also analog of 6-mercaptopurine. This molecule have immunosuppressive activity and it was discovered in 1957. But few scientists actually find another analog of this molecule 6-mercaptopurine that is called azathioprine, and they received Nobel Prize. This molecule prevent DNA synthesis and RNA synthesis, that means replication and transcriptions. So it is actually mitotic inhibitor. So it inhibits mitosis. Cyclophosphamide. This is actually a alkylating agent. This agent actually cross-link with DNA and also prevent DNA replication and RNA synthesis, that means transcriptions. Okay, perform the same function. So it is also mitotic inhibitor. Another example of immunosuppressive drug is methotrexate. This methotrexate is actually antagonist of folic acid. And what is the role of folic acid? Folic acid actually helps to synthesize purine and pyrimidine base, DNA and RNA synthesis. The same function is performed by this methotrexate. Another immunosuppressive drug example is mycophenolate mofetil. It is also known as MMF, mycophenolate mofetil. This drug actually inhibit directly guanine synthesis, and they actually preventing DNA synthesis and RNA synthesis. So these are called, these 4 type of medicine are actually called anti-metabolite. Now I will show you another form of immunosuppressive drug that is called anti-inflammatory corticosteroid. Example is prednisone, dexamethasone. Fungal metabolites, fungi, from the fungi it is derived. Example is cyclosporin A, also known as CSA, and tacrolimus, also known as FK506. Cyclosporin A, this medicine or drug actually when incorporated or actually enter into the cell, they actually bind with a molecule that is ubiquitous in all cells, that is called cyclophilin or cyclophilin. They bind with this molecule, and both this molecule cyclosporin A and cyclophilin actually inhibit a molecule called calcineurin. And what is the role of calcineurin? Calcineurin actually activate a transcription factor called NFAT, and this NFAT actually activate or helps to transcribe 2 major important gene, that is called IL-2 and another is IL-2 receptor alpha, that means CD25. In my T-cell activation class, I have discussed about the role of IL-2 and CD25, that means the IL-2 receptor alpha segment subunit. So when this cyclosporin inhibit this calcineurin, then this total chain reaction stop. Tacrolimus is another molecule called FK506. This molecule also bind with another partner called FKBP, that means FK binding protein. This molecule when actually bind with each other, they also perform the same function like this cyclosporin A. They also inhibit the calcineurin. Rapamycin. So this rapamycin actually inhibit a enzyme that is called mTOR. mTOR means mammalian target of rapamycin. This enzyme is very important for a signaling that is called AKT signaling. This signaling actually helps in the survival and proliferations. That means this mTOR actually activate or actually promote proliferation. So when rapamycin inhibit this enzyme, proliferation also not occur. This rapamycin is also known as sirolimus. Monoclonal antibody. In my previous class, I have discussed that this rejection of the transplantation actually performed by alloreactive T cells. So T cell is very important. So this monoclonal antibody are actually generated in response to prevent these alloreactive T cells. Let's assume this is the T cells, alloreactive T cells. That means these cells can recognize allogenic MHC molecules. As we all know that a T cells must have a T cell receptor, And as we know that T cell receptor is associated with some co-associated molecule for the signaling, that is called CD3. For the proper activation of T cells, there is another important thing that is called a receptor called CD28. So for the proliferation of T cells, there is another molecule or receptor that is called CD25. This is the IL-2 receptor alpha subunit. These responder cells are actually showing allogenic MHC molecules. Let's assume this is the dendritic cells of the donor. They can show some co-stimulatory molecule like CD80 or CD86 that can bind with this CD28. Another molecule is actually responsible for the activations of these cells, that is called CD40L. Antibody against the CD3 protein. Why? Because CD3 helps in the T cell receptor signaling. So this is the first signal from the T cell receptor, and CD3 helps in the signaling. They use antibody, monoclonal antibody against the CD3 molecule. They name it OKT3. Not only that, they also added diphtheria toxin on this antibody. So when any T cells bind with this antibody, they actually, this diphtheria toxin actually enter into the cell and actually kill the cell. Another thing, they use anti-CD25 antibody, monoclonal antibody against this CD25. They also used another molecule that actually prevent this signaling, that is the second signaling. They use CTLA4. This is also a CTLA4, but with a antibody, tagged with antibody. So this is against the CD80 and CD86. So this molecule can be bind with CTLA4 with IgG antibody fusion. When the CTLA4 bind with this CD80-86, this CD28 cannot bind with CD80 and 86. They are prevented. So this signaling can be prevented. Antibody against the CD40L. They try to prevent any sort of signaling. So they discover CD40 ligand. And I want to share that there are also some side effects of this monoclonal antibody. Not only that, when T cell receptor actually activated, they actually start a pathway that activate calcineurin, that activate NFAT, that actually helps to transcribe IL-2, CD25. Now this IL-2 molecule actually produced by these cells itself. This is IL-2 molecules, cytokine, that actually bind with this CD25 and helps to proliferations. So for this reason, scientists have used inhibitor of this calcineurin like cyclosporin A, tacrolimus means FK506. And another thing is, when this IL-2 bind with this IL-2 receptor, that means CD25, another signaling starts, that is called mTOR signaling. So mTOR actually activated, and for that reason scientists used rapamycin to prevent this mTOR. So this prevent calcineurin. So this total chain reaction is stopped, and this rapamycin stop the mTOR pathway, and mTOR actually helps in the proliferations, because mTOR help in protein synthesis, and actually survival and proliferations prevented. I have mentioned that azathioprine, cyclophosphamide, methotrexate and mycophenolate mofetil, all these molecules can prevent the cell cycle because they prevent the formation of DNA and RNA.